Hello everyone, welcome back. Chapter 10, Part 2. We're going to be talking about the renin-angiotensin aldosterone system, as well as ADH and fluid balance, and then finishing up the rest of Chapter 10, which goes very quickly. We've talked about this before. We talked about this in the Chapter 5 lesson, I think. We talked about this, but we should do it again, because people forget this all the time. And also, I have a supplemental video about the renin-angiotensin aldosterone system, but now we know where it works, right? So we talked about this diagram of the nephron that we had before, the PCT, loop of Henle, little baby nephron, right? DCT, blah, blah. Aldosterone is functioning right here. So the kickoff for the renin-angiotensin aldosterone system is, number one, low blood delivery to the nephron is detected as low blood pressure. By the way, I made a mistake in the last video. Reabsorption is when you take things in the lumen and bring them back into the bloodstream. Secretion is taking things from the cells of the lumen or in the bloodstream and sending them into the lumen. So the bloodstream sends stuff in, that's secretion. Or the cells send stuff in, that's secretion. Things coming out of the lumen is reabsorption. So reabsorption is in to out, and secretion is out to in. And I told you to not worry about when things go in, so do not worry about secretion. Except for this example we're about to do on the board. This is the one that you need to know. Guys, there's a big thing. You guys know Sheikh A.N.J., Qadam, Sheikh Abdul Nasser, right? He has a quote. He says, "We have rules and we have exceptions." We have rules and we have exceptions. Do not make rules out of the exceptions. I don't have to make wudu on my broken leg that's wrapped in a cast. I don't have to do that. Does that mean I never have to make wudu? No. It's not true. If I have a bandage on my finger, what do? I take it off, right? Doesn't mean I can wipe over everything. Oh, my hair looks so good today, I'm just going to keep my hat on. No, take your hat off. We don't make rules out of exceptions, but we have exceptions to rules. So, decreased blood delivery to the nephron is detected as low blood pressure. Juxtaglomerular cells, which now means something to you, the cells next to the glomerulus, that bundle of crazy wires that delivers blood into the nephron, juxtaglomerular cells secrete, release renin, which is an enzyme. Do you guys know why I say renin is an enzyme every single time? Because I need you to remember what it does. An enzyme does something. It cleaves the next molecule. 3, renin cleaves angiotensinogen into angiotensin 1. There's no such thing as angiotensin. Angiotensin 1. Is angiotensin 1 active or inactive? Inactive. Which is why, despite the O-gen ending of angiotensinogen, meaning that it's a zymogen, I previously said that a zymogen is a molecule that must be cleaved to be activated. This is an exception. So that means that not all zymogens are activated when they're cleaved. So I since edited my statement. I said a zymogen is a molecule that must be cleaved in order to move forward, in order to move the process forward. Or a zymogen is a molecule that must be cleaved. Angiotensin 1. 4. Angiotensin 1 travels to the lungs, is acted upon by greatest enzyme name of all time. What is it? Angiotensin converting enzyme. Angiotensin converting enzyme. If only everything were that simple. Actually, it is. And I'll explain why it's that simple at the end. And it becomes, so to become angiotensin 2. Who said Ang2? You worked in a hospital before?
No. I'm in farm, and so I'm
Gotcha. Angiotensin 2. Angiotensin 2, active or inactive? Active. There's too many words. Let's start shortening things. Angiotensin 2 leads to increased heart rate, increased vasoconstriction. Increased vasoconstriction just means decreased radius of the blood vessels, right? And it will hit the adrenal what? Adrenal cortex. Cortex, salt, sugar, sex. You're looking for the salt portion of that. To secrete aldosterone. So we do this whole thing just to get to the thing that does something here. Huh? Vasoconstriction. That's too short for me.
So is angiotensin just like floating in the blood?
Yes. It's made by the liver and just floats around. Aldosterone increases activity of sodium, potassium, ATPase, not pumps, ATPase, to reabsorb 3 sodium and secrete 2 potassium. Water follows the sodium, potassium, sodium. Since they're being reabsorbed, that means the sodium and the water enters the vasa recta and into the bloodstream. So the effect of aldosterone is increased sodium reabsorption, increased water reabsorption, increased potassium secretion, which is potassium diuresis, meaning we urinate out our potassium. Yes? Good. And that all happens here in the distal convoluted tubule. Do you guys see that? Do you guys also see how the blood pressure is equal to the cardiac output multiplied by the systemic vascular resistance, which means that the blood pressure equals the heart rate times the stroke volume times the systemic vascular resistance. Number 1 and number 3 are increased by angiotensin 2, and number 2 right here is increased by aldosterone because of the increase of the water coming back into the bloodstream. So if you're increasing the blood volume and you're trying to keep the circulation the same, you have to increase the stroke volume. So those 3 will increase the blood pressure. But all of those come after the conversion of angiotensin 1 to angiotensin 2. So if you were to block angiotensin converting enzyme, you could stop all of that from happening, and this is the basis of the blood pressure medication ACE inhibitors. An ACE inhibitor will inhibit the conversion of angiotensin 1 into angiotensin 2, and since you don't have angiotensin 2, you don't get the increase in the heart rate, you don't get the vasoconstriction, you don't get the release of aldosterone, you don't get the potassium diuresis and sodium reabsorption, you don't get the increase in the stroke volume, and you don't get the concomitant increase in blood pressure. Does that make sense? Very effective medications. But they have since been replaced. ACE inhibitors include enalapril and captopril. They have since been replaced by angiotensin receptor blockers, or ARBs. And these medications include valsartan, Your sartans, that ending means angiotensin receptor blocker. What's the difference between ASI and angiotensin receptor blocker? One of them blocks angiotensin 2, one of them blocks angiotensin converting enzyme. They basically do the same thing. It's just that ACE inhibitors cause a nasty cough. ACE inhibitors cause a nasty dry cough, and the ARB doesn't. That's it. Why does it cause a cough? Because you're in the lung. ACE is in the lung, so your side effect is respiratory. Yes?
What is standard NF?
Huh?
What does aldosterone do?
H2O plus, H2O reabsorption and potassium secretion. So back into the lumen. This is the one time you have to remember that, and the potassium goes out in the urine. Yes?
How does the angiotensin 2 affect stroke volume again?
Well, angiotensin 2 doesn't directly affect stroke volume. Aldosterone affects stroke volume.
Aldosterone, sorry.
So aldosterone will increase the activity of sodium-potassium pumps in the distal convoluted tubule. Sodium goes out, potassium comes in. So the exchange rate here actually is 3 sodium goes out, 2 potassium goes in, and since there's more charges going out than coming in, the water follows more of the charges, it goes out.
Okay.
And remember that in this process, ATP is hydrolyzed. We're going to have a long discussion about ATP hydrolysis. A long one. And about why these carboxylic acid derivatives with multiple phosphate groups and these phosphor anhydride bonds and the hydrolysis of these bonds and how that induces charge separation within the phosphate groups that allows for the removal of like charges, which decreases the thermodynamic instability within that localized region. We'll talk all about it. We will. We will, we will. You will know about ATP hydrolysis better than anyone in this building other than a PhD. Yes?
Is there a reason kind of like why the process is so, I guess, multi-steppy in a sense?
I've talked about this before. Why is anything in the body in multiple steps? Amplification. One molecule of renin cleaves 10 angiotensinogens, makes 10 angiotensin 1s, makes 10 angiotensin 2. 10 angiotensin 2 releases 100 aldosterone molecules, all for amplification. Yes?
I know you said that like all of this happens in the distal convoluted tubule. But,
Yep.
When we detect low blood pressure, that's by the actual glomeruli.
Yeah, so that's in a bundle of cells that's actually like right here. And you're not going to know it, but the cells aren't right there per se. The nephron actually sort of loops in on itself. This is a very simplified diagram. The nephron actually grows and then twists onto itself. So the DCT and the PCT are actually like sort of right next to one another, and then the juxtaglomerular apparatus is somewhere within there. Yes?
What happens in the distal convoluted tubule?
The same thing that happens in the PCT plus aldosterone. Some people like remembering this little mnemonic that's on like every Anki card ever, that your waste products in nephron physiology are what? Hunk, H+, urea, NH3, and K+. Oh, should I do my urea discussion? It's like a biochem discussion, but I really want to do it. Okay. All right. Yeah, of course, 100%. You think I wouldn't go over something that makes me this excited? Let's talk about urea for a moment, because urea is a beautiful thing. All right. Who here has not taken organic chemistry? Beautiful. Oh, that's just beautiful. All right. What is the hybridization of that nitrogen? sp3. I heard sp2. I heard sp4. Wow, that's a new one. We're reinventing the wheel here. Yeah, let's just keep going. sp69. I mean, yeah, sp67. Yep, 100%. All right, your choices are sp3 and sp2. Who says 2? We got 3 people. Who says 3? We got like the whole rest of the room. It's sp2. Why is it sp2? Yes. Guys, that nitrogen resonates. You should remember from organic chemistry that nitrogen lone pair adjacent to an sp2 carbon will always resonate. What do the resonance structures look like here? We have NH2 lone pair. This is an sp2 carbon. Resonates its electrons up towards the carbonyl group, creates a negative charge in the oxygen and a positive charge on the nitrogen. You would assume that this is incredibly unstable. However, this is actually like a 60-40 mix. With anything else, it would be wildly unstable. This is the exception. We have rules, we have exceptions. We do not make rules of exceptions. This is an exception. Now, if this didn't happen, your DNA wouldn't fold in on itself properly. We will discuss that when we get to biochem. However, in your body, when you break down amino acids, do you guys see how much more I love chemistry than biology? Your amino acids will break down into NH3/NH4, ammonia/ammonium. Yes? This is a weak base, and this is a weak acid. The weak acid with a pKa of approximately 9.4. Right? So if I were to put you on that on a scale for you, you have carboxylic acids at around a 4 to 6. You have carbonic acid at around a 6.2. You have RSH, which is around like an 8. You have HCN, which is around a 10. You have water, which is like a 15.7. And you've got NH4 hanging out like right around here. So you're sort of in that like weak acid range, just coming close to water. So it's like almost neutral, but not really, because it's still like 5 points higher than that, right? Because remember, a lower pKa means more acidic. And it's more acidic by what degree 1 point is times 10. There's a logarithmic scale, right? So much less acidic than a carboxylic acid, but still equally more acidic than water, right? And that NH3, of course, is a weak base. So if you let this thing go free, it's going to start protonating stuff. And if you let this thing go free, it's going to start deprotonating stuff. So you need a safe way to take this and send them throughout the body. Also, ammonia is a neurotoxin. If you guys would like to look at what ammonia does in the body, you can look up this specific sign called asterixis. And you can also look up hyperammonemic encephalitis to see the differences that ammonium makes inside of the central nervous system. So in order to transport this and urinate this out, we need to make it sort of palatable to the rest of the body. What we do with that is we take two of these molecules and we just link them together. We take one and we take another. And you're going to say, Yusuf, this doesn't solve your problem. Those still both have lone pairs on them. So what do? We conjugate them to something that they can resonate with. And that is known as urea. So now this is resonating, and that's resonating. And since this whole thing is resonating, you now have a distribution of those lone pairs, and it's much less reactive than before. And you can safely transport this throughout the body without destroying everything in your path. Very cool. Yes?
Random question. When we do if it's like a weak acid and it's like the conjugate, why is it not like a strong base?
Because that's a misnomer. Strong acids produce negligible bases. Strong acids, the conjugate acid will be a negligible base. A weak acid will produce a weak base. A negligible acid, if you were to deprotonate it, would create a strong base. Think of strong, give me a strong base. Anyone? HBr. That's super weak. OH minus. What's the conjugate acid OH minus H2O? Is that an acid? No, it's a negligible acid, produces a strong base. Give me another one. Huh? HCl is a strong acid. HCl is a strong acid, so it produces a negligible base. Next. HBr. HBr is a ridiculously strong acid, produces Br minus, a negligible base. In organic chemistry, we'll go over why these are negligible and why they don't react with anything. I want you to know the difference between this and this, and why this one reacts more than that one. Huh? Did you say something? Okay. Guys, I love chemistry. I love chemistry, which means you are going to absolutely hate it. You're going to hate it so much. But you're going to learn a lot. But you're going to, like, does anyone feel tired when they walk out of here? Like when we're done with the whole lecture and everything like that, you guys have been here for like a couple hours. Are you guys tired? Because I'm tired. You guys are going to be very tired. I'm going to make you write a lot. I would quite literally suggest you get used to drawing your molecular diagrams. Like if you want to start practicing your molecular diagrams, I would advise it. I think it's advisable to sit there and copy down molecules that you think are important. Because the one question I get asked every single time is how do you draw your molecules that fast? Well, I took organic chemistry almost 7 years ago now, and I've been drawing molecules since. On the topic of fluid balance, we have another hormone that is very easy to understand. Antidiuretic hormone. Antidiuretic means it stops you from peeing, correct? Coffee is a diuretic. Caffeine is a diuretic. Right? When blood osmolarity is high, hypothalamus sends a nervous signal to the posterior pituitary. Very good, because I said nervous signal. Guys, shut up. Sends a signal to the posterior pituitary. The posterior pituitary secretes ADH, which goes to the collecting duct. And the effect is increasing the number of aquaporin channels in the collecting duct to increase water reabsorption. And this leads to an increase in blood volume. Let's go over osmoles again. Molarity equals moles over liters. So ADH says when the molarity is too high, I'm going to bring in more volume to bring this back down. Yes? Good. Aldosterone brings in salts and volume. So what happens to the molarity? We actually don't know. It depends on the volume status of the patient. You'll hear this word a lot in medicine, volume status. You ever heard anyone describe a patient as dry? It means they're super volume down. No. Why? Why would salt follow ADH? It's literally just a hole. You put a hole in the collecting duct and water gets out. It's called an aquaporin. It is a pore for water and nothing else.
So is there some sort of mechanism that's preventing the salt?
There is a pore for water. Why are you asking about salt? It is not a pore for, it is an aquaporin. Okay, look, this is the collecting duct. You put a hole in it and the hole says Water. And you guys are like, what happens to the salt? Shut up. It's right there. It says water. Water. Salt? No, we talked about this. That's aldosterone. Shut up. Enough about the salt. Yes.
So is it water specific or is it?
I'm gonna guess.
I was asking more, what's the mechanism that prevents the salt from leaving the...
Because it's water. What do you mean what prevents it? It's for water. Okay. You know what? When we get to biochem, we are going to say that insulin activates GLUT4 channels for glucose. You guys are gonna be like, yeah, but what about the salt? What the hell? Get out. Like, actually, just get out. See me after class. You guys are weird, man. You guys are some wacky characters. I feel like if someone took like the Smiling Friends cast and just threw them in my freaking hall. And it was like, here, teach these idiots. Not you guys in the back, just like everyone up here. Except for you. I know every single one of these people personally. Yes.
I have a question. So since caffeine is a diuretic, does it prevent the ADH from binding or to cause more aquaporin?
Ooh, okay. So the intimate relationship between like caffeine and diuresis is like pretty complicated and beyond like the spectrum of what you would need to know. But I would assume that yes, there is some degree of blocking of ADH.
Okay.
Yeah. Why is ADH also called vasopressin? There's something on the board that's telling you why it's called vasopressin, because like vasopressin like pressurize the vessels, right? Blood pressure. So what's on the board that might actually increase the blood pressure? Yeah, the blood volume. That's a lot of volume right there. In the form of water. So sometimes when people are in like complex traumas, we'll actually shoot them full of vasopressin to like get the kidneys to immediately start bringing in the fluid that they have inside of them so that we can artificially increase their blood pressure. One more thing that's actually pretty interesting is that sometimes when you have a pregnant patient who is undergoing uterine atony, which is basically when the uterus doesn't like contract fully after the baby's delivered, you shoot the uterus full of vasopressin. Why? Because oxytocin and vasopressin come from the same part in the pituitary. They share a very similar structure. So oxytocin does this, vasopressin does the same thing as oxytocin, where it causes the uterus to contract. So and the thing is, when I say you shoot the uterus with vasopressin, you take a needle, put it in the uterus, and shoot it with vasopressin. The last thing in chapter 10 is the skin, the integumentary system. Just call it the skin. I can like feel my blood pressure. I can't do it. I just can't do it, man. Skin is the what organ in our body? Largest, right? It's about 16% of the body weight on average is skin, right? We have the structure of the skin. So first we have the epidermis, which is the uppermost layer of the skin. It's split into other layers called strata. The first layer is the stratum what? Anyone? Corneum, I believe. It is a stratum corneum, right? Also called the horny cell layer, literally. And this is just layers of dead skin. Layers of dead skin cells. That's what we have to get through to get to anything deeper. You have a special layer after this called the stratum lucidum. I put it off to the side. which is present in thick hairless skin. So this is present in the palms of hand and where else? Soles of feet. Don't be weird. Yes?
Is that like would calluses be just be extra thick and loosening?
I think so. I don't know. Ask a dermatologist.
You didn't have a derm rotation?
Huh?
You didn't have a derm rotation?
No. Why would I have a mandatory rotation in a subspecialty of like a specific branch of surgery? You only do a derm rotation if you want to. So what are the mandatory rotations in medical school? In the order that I did them. Well, you guys wouldn't know. Psych, neuro, OBGYN, psych, neuro, peds, OBGYN, general surgery, anesthesia, elective surgery, internal medicine, family medicine, radiology, emergency medicine, and research. And then you do your sub-Is and this and that and everything. You need a sub-I in medicine. You need a second, at my school, you need a second round of family medicine, which is usually geriatric psych. And then you decide on your electives and this and that and blah, blah, blah. Yes?
What rotation did you enjoy the most?
Did I enjoy the most? Obviously, internal medicine.
It wasn't OBGYN?
OBGYN was the most intense and the most informative. I'm going to kill him.
Go ahead.
I'm going to kill him.
I thought your back liked it.
Oh, because I herniated I did herniate my disc during my OBGYN rotation. I did. I woke up one morning, I put my foot on the ground, and I was just shooting pain. I was just like paralyzed on half my body. It took me like 5 or 10 minutes to get up off my floor that morning. And then I went to the ER. They gave me Toradol.
Nice.
That was good stuff. That was good stuff. And the thing is, it's like the weakest opioid ever, but it felt so good.
Is it an opioid?
Minor. It is a major and
Toradol is Toradol.
it is a major NSAID. Sorry. They didn't give me Toradol. They gave me Tramadol. Yeah, they gave me Tramadol. So they gave me it's like a major NSAID and a weak opioid agonist.
Runner's high or Tramadol?
Runner's high. Nothing beats Runner's high is the best thing I've ever felt in my entire life.
What does it feel like?
What does it feel like? It's something I'm not allowed to say on camera.
Oh, okay.
Okay, so the stratum granulosum comes after. And this is the layer where these cells actually die and lose their nuclei. So keratinocyte, which is the type of layer, the cell that's up there as well. Death and denucleation. Denucleation meaning removal of a nucleus. In the stratum spinosum, wow, my handwriting is so bad right now. We're doing so good. In the stratum, that doesn't say stratum. We're going back to the thick marker. As if that's going to make a difference. The stratum spinosum the keratinocytes link together and in the stratum basale, the basal layer, you have keratinocyte proliferation. You guys have heard of basal cell carcinoma, right? The basal cells that replicate, they replicate too much, and you get basal cell carcinoma. Deadly or not? Not really. It's really slow growing, usually caught when it's not really that advanced. You're usually just able to excise it and you're good. Squamous cell carcinoma is a more aggressive version of basal cell carcinoma that can kill you, and the worst skin cancer is melanoma. So keratinocytes Keratinocytes produce keratin for strength and structure. You're asking that the keratin is actually what causes the calluses when you injure the skin. It's an excess of keratin in that place. You have my favorite type of skin cell. What is it? Melanocyte. Because I am brown and beautiful. Produces melanin, which is pigment inside of your skin. Does anyone know what type of, that's a very high level question. Never mind. It's like a USMLE question. I'm asking you about the most common cancer biomarker for melanoma. It's like there's specific types of melanoma and they can be differentiated by one specific type of biomarker. It's called S100. S100 is a biomarker for melanoma. If you guys ever worked in a derm lab, you've probably heard that thrown around before. And we've talked about macrophages inside the skin. What are they called? Langerhans cells. Dendritic cells. Yeah, dendritic cells are like those sort of like different. They're in that in-between area, I believe. If I'm not mistaken. I don't know. I don't remember. The dermis. The dermis is below the epidermis. Yes?
Are the Langerhans cells in the stratum granum? Or which layer were they in?
I have no idea.
Oh, okay, never mind.
I don't know where they are. I wish I knew where they were. The question was, the Langerhans cells are in what layer? I don't know. The dermis, which is below the epidermis, is split into two layers. You have a papillary layer, and you have a reticular layer. This is where your sweat glands blood vessels and hair follicles originate. And you also have a bunch of sensory receptors here. There's a couple of them. Did I ever memorize them? No. You have the Merkel cells. I think they're like Merkel cells. You have the Meissner's corpuscles. You have one more. You have the Ruffini endings. And I think you also have the Pacinian corpuscles, right? Pacinian corpuscle is such a weird word. Corpuscle is such a weird word. Okay. The Merkel cells detect deep pressure and texture. Don't be weird. The Meissner's corpuscles are my favorite because they detect what? What is it? Let me check. Light touch. Sorry, they're not my favorite. There's a different one that's my favorite. Light touch. The Ruffini endings will do stretch. And the Pacinian corpuscles are my favorite because they do deep pressure and vibration. And the way that they do vibration is actually interesting. If you look at a Pacinian corpuscle, if you zoom in on it, it actually kind of looks like a little spiral. You'll see it actually looks like this. Someone look up the histology of a Pacinian corpuscle. And it literally looks like it has a little ball on it that'll vibrate when vibration hits the skin. Pacinian corpuscle histology is literally like the cell wraps itself like that.
It looks
Yeah, look, it's pretty cool. Looks cool. Yeah.
Do you have any smart way to memorize these? Other
No.
than this group?
Why do you think I didn't memorize them? Because I'm not smart.
I hated that part.
The last thing about the skin is thermoregulation. Hot equals sweat. Cold equals shiver and goosebumps to make hair stand up. What does sweating do for the body? Yeah, basically what happens is that the atmosphere around you is so hot that your body sends out water as a protective barrier against your skin. And since water has such a high specific heat capacity, all that heat in the environment goes towards evaporating the water off of you instead of burning your skin. That's how sweat works. What does shivering do? Contraction of skeletal muscle breaks ATP. With that contraction, breaking of ATP releases energy. Energy gets released as heat, keeps your body warm. And what about the hair standing up? Protects against wind, things like that. Yes? That's the end of chapter 10. That's it. Now, let me take a look, because I'm not teaching chapter 11. I don't feel like it. What time is it?
6:38.
We will do chapter 12 and be done. Thank you guys so much for watching. See you in the next video.